What's up, guys? Today we're going to be talking about operons, or more specifically, the Lac Operon. So an operon is essentially a cluster of genes that are transcribed together, and these operons only occur in prokaryotic DNA. So here we have a prokaryotic strand of DNA in our operon setup here. So there are several parts to every operon. So first of all, we have the promoter here. So the promoter is it's the space that allows RNA polymerase to bind onto the DNA here. So that's important for when we need the RNA polymerase to transcribe our genes that we're trying to express. So after that, we have the operator here. So the operator is essentially a gate for the RNA polymerase. So here we have our structural genes, which are it's the information that we're trying to express. And our operator here is kind of safeguarding the genes, so we can regulate when they are expressed. And then finally, we have our regulator. So our regulator gene produces a regulator to open and close our gate to let the RNA polymerase through. So an operon can be either inducible or repressible. In an inducible operon, it is off by default. So, and it can be turned on by an inducer. But so to be off by default, we have our regulator produced by the regulator gene, and it is always attached to this operator, right? So while the regulator is attached, our operator gate is closed, and RNA polymerase cannot move past this point to get to the genes it wants to transcribe. And so in repressible operons, which are on by default, our regulator is absent until an inducer comes along and allows the regulator to bind to the operator to stop the gene expression. So in our specifically Lac Operon, we're trying to digest lactose, right? So it's an inducible operon, and lactose is a notoriously complex sugar, right? So the genes that we're trying to express here are all responsible for aiding in the metabolism and the digestion of the complex lactose sugar. All right, so yeah, we'll set our regulator up here for the start. And so for lactose, that brings along our inducer. So our inducer, of course, is lactose. Because we're digesting lactose, we need the presence of lactose in order to activate the Lac Operon. And so lactose is converted into our allolactose here, which is the molecule that will bind to the regulator. So this is allosteric inhibition. So the allolactase will bind to the regulator and kind of mold it out of shape so it won't fit into the operator anymore. And so this will cause the regulator to fall off and the operator will be uninhibited. And it's pretty easy to remember the inducer here, because since we're allosterically removing our regulator, we need allolactase, right? Okay, so with this, our regulator is out of the way and the operator gate is opened for our RNA polymerase to move along and start transcribing our structural genes. And so the transcribed mRNA will then move along to translation to be converted into proteins. And that about sums up the Lac Operon's process. And it can get a little more complicated if we factor in the presence of glucose instead of glucose in addition to lactose in this process, but that could be a topic for another video. So thank you guys so much for watching.